And now we're going to move on to our talk by Dr. Natalia Fedianina from Beckman Coulter, who's going to discuss the use of their flow cytometer and how that can be applied into studying muscle health in general. Natalia?
Thank you very much. Can you hear me?
Yes, and we can see your slides too.
Okay, can you see me? I cannot see myself for some reason, but anyways, that's fine. I don't know why it's not working. I hope I don't break off during my presentation. Okay, let's start. So my name is Natalia Fedianina, and I'm a flow cytometry consultant for Beckman Coulter. I'm also a huge nerd when it comes to exploring different methods in life science research. So, for example, when I was doing my undergraduate studies at York University, I was using eye tracking technology to research eye and hand movement coordination. Then in graduate school, I did patch clamp electrophysiology. I think a majority of people who tried to do patch clamp can appreciate even more the simplicity and power presented by flow cytometry. So I want to thank Dr. Hood for a chance to talk about the versatility of flow cytometry at this conference. Okay, sorry, I don't know what's happening. I cannot, just slow. Okay, perfect. So I hope this presentation will give you some ideas about how this technique can help you in your research. I will touch upon flow cytometry basics and the muscle health related flow cytometry applications I came across during my literature review. So why use a flow cytometer when there are microscopes, spectrophotometers, plate readers, Western blot, and other methods of cell characterization? Well, similarly to fluorescent microscopy, you can study multiple parameters at a time for each cell. Expression of multiple biomarkers, granularity, and size. Also, like microscopy, and unlike plate reader technology, it allows you to analyze each cell individually with great sensitivity. Unlike microscopy, it allows you to analyze tens of thousands of cells per second. Here is a bit of background. So the principle of flow cytometry is simple. You line up the cells in a stream of fluid, one cell at a time, and one cell at a time is passing in front of a laser or lasers. The forward and side scatter is recorded, and this gives researchers some idea about size, granularity, and shape of the interrogated cell. The laser of a specific wavelength excites a fluorophore on a cell to emit a fluorescent signal, and this signal is recorded by detectors. In the ideal experiment, each detector would record light from one fluorophore. So, for example, on a 13-color flow cytometer, we could interrogate the same cell for the presence of 13 different biomarkers, which potentially could all be colocalized on the same cell. One of the most straightforward applications of flow cytometry in every field is the absolute cell count, as well as the enumeration of live and dead cells. For example, it could be used for counting of mononuclear muscle cells in culture. Some newer instruments on the market, like Cytoflex, no longer require the use of beads to count cells. The cells could be counted directly by counting the number of fluorescent events. Live and dead stains could be used to count live and dead cells. And some typical probes would stain dead cells, and live cells would remain impermeable. But for example, such a dye as mito-633, on the opposite, would stain mitochondria in live cells, and the staining is lost when mitochondria become depolarized during cell death, apoptosis. Apoptosis or cell death could be one of the signs of disease and muscle atrophy. And there was a lot said about muscle atrophy in earlier talks. Loss of the mitochondrial membrane potential is a sign of apoptosis. In one study, the membrane potential was investigated to explore differences between mitochondria in muscle cells in young and senescent animals. So JC1 is one dye commonly used for these studies, and it has an interesting property. So as a monomer, it emits green fluorescence, and as an aggregate, emits a red fluorescent signal. So JC1 aggregates in polarized energized mitochondria, and it presents itself as a monomer in depolarized, de-energized mitochondria. So this property of JC1 was used to find that in old rats, subcircular mitochondria have a nearly double reduction of mitochondrial membrane potential. As I was doing this literature review, I noted that Perlman et al. Suggested an improvement to the existing use of JC1. They recommended exciting the dye by 4 or 5 nanometer violet laser versus a typical 488 nanometer laser. This produces less spillover from JC1 monomer and minimizes the need for compensation and makes the data more accurate. Studies of calcium flux in various cell types is one of the well-known flow cytometry applications. Calcium release from the sarcoplasmic reticulum into the cytosol causes muscle contraction, and release of calcium into the cytosol is essential for muscle contraction, and then reabsorption is essential for relaxation. Adding ATP to muscle cells results in transient release of calcium in calcium flux type of experiments. And this could be recorded using such dyes as fluor3 or fluor4, which bind to calcium. There is one feature available only on Beckman Coulter's Cytoflex flow cytometer that could enhance such studies. You can add an agent to trigger calcium flux right at the time when the sample is aspirated into the fluid stream for interrogation by the laser. So as you can see in the picture on the right. In other flow cytometers, you need to record the baseline, then stop the recording, add the calcium flux agonist, and then record and then restart the recording, which means that some early events might be missed by the time the sample is aspirated for analysis, and some desensitization could already occur. A hot subject in cellular and systems biology is the power of exosomes. And I saw there was a mentioning by one of the earlier presenters of some exosomes they were doing. So I also saw in early publications in which a flow cytometer was used to analyze content delivery via exosomes to muscle cells. Exosomes were labeled with PKH67, and this labeling allowed to visualize exosome incorporation in muscle cells. Beckman Coulter's Cytoflex flow cytometer could also be used for such studies, but is also uniquely sensitive to allow for detection and quantification of isolated exosomes. Its stated limit of detection right now is 80 nanometers, and the ability to detect and quantify exosomes directly via flow cytometry became possible in the last 3-4 years. So I hope it will open the door to further investigation of exosomes for both pure research and for applied purposes. One interesting tool for biomarker discovery that was launched recently is the layer plate screening assay. The assay consists of a set of 96 well plates in which each well contains an antibody against one surface marker. This allows to test against the presence of hundreds of antigens in one run. The screen could allow to identify expression of known proteins on myoblasts, for example, or fibroblasts, macrophages, among others. And this could also help analyze isolated targeted cells from heterogeneous populations, or it could be used to compare protein expression in different stages of cell cycle or under different cell culture conditions. Among the papers I reviewed, there were quite a few other interesting applications such as reactive oxygen species, macrophages, Tregs, muscle stem cells, and autophagy, of course, in muscle injury and regeneration. Unfortunately, my time slot is too short to cover everything I'd like to cover. I know that this review is far from being exhaustive, but I hope I give you a good sense of the power of this technique. In the span of time, a patch clamp might answer one experimental question flow cytometry would answer tens or hundreds. No offense to batch clamp physiologists present. I was one myself. One last thing. So we have a great tool to enable muscle health research. Beckman Coulter's Cytoflex is an industry-leading flow cytometer. Although it has been on the market for only 6 years, it became wildly popular, and there are more than 40 Cytoflexes in the Greater Toronto area alone. In designing this flow cytometer, engineers used new detector technologies like APDs versus traditional PMTs, and new integrated optics and new lasers. And all of this resulted in a very sensitive instrument, one of the most sensitive on the market. This sensitivity would be especially useful for dim signals, intracellular probes, or weak expression. It's also useful for detection of small particles like exosomes. Cytoflex can detect fluorescence from all the known dyes for flow cytometry. It can analyze up to 21 colors at a time, which could be useful for all those immunophenotyping studies. It allows for beadless volumetric control and volumetric absolute count and particle counting. And it has a flexible sample uptake, which becomes useful for calcium flux studies that I mentioned earlier. And it is compatible with both tubes and plates, which is required for screening assays. I hope you enjoyed this talk. Thank you very much for your time. I will be very happy to answer any questions you might have. Also, please feel free to reach out to me via chat, email, or LinkedIn.
Thanks, Dr. Fedianina. That was a good overview on important applications. Does anyone have a question regarding this talk? You can.
Natalia, thank you very much. That was very instructive for us. And I just wondered how you've described a number of applications which are very useful. I wonder how does the instrument handle a myotube? We talk about myoblasts and single cells all the time. How is a myotube too big for that instrument, or will it work?
No, I have seen publications that use myotubes. Of course, it depends on the size and how big the myotube is. But that's something that, again, I saw publications, so it's definitely doable. So the best way to find out is to test.
Is to test it, yeah. Test it, plug it up, and then go back.
Do that during the demo, period.
Do that during the demo, yeah. Yeah.
Exactly.
I was going to ask how big the flow cell is, because that was going to be my question about myotube as well. Just like
actually, it's a great question. So yeah, it depends. Of course, there are some instruments that clog easily, and then if they aspirate, they stop working, and it's a huge pain. But with Cytoflex, from what I have seen and heard from my colleagues, it does not clog. So my understanding is that I don't have an exact diameter for you right now. I will look it up. But so far, I have heard that it is great when it comes to clogging. It really differentiates itself in that sense.
Thank you. That's helpful to me.
Okay, thanks, Dr. Fedianina, and thanks everyone. This was a very, I think, instructive session. And